Hi, in our last discussion, we realized that there are many potential costs to sexual reproduction, and yet it turns out that sexual reproduction is extremely ubiquitous, widespread in the living world. So, that led us to ask the question that what are the benefits of sexual reproduction, and as I said that several theories have been put forward. So, in this particular discussion we are going to discuss about 3 of those. Now, the first and the most important one that comes straight away to your mind when you think about the benefits of sex is in terms of generation of novel genotypes. Why is that so? That is because, remember, whenever there is crossing over happening during meiosis, then alleles are actually coming together in all kinds of new combinations, and therefore, whenever there is epistatic variation that is happening, that is variation due to the identity of the alleles in various loci, then all these new combinations are going to throw up all kinds of variation at the phenotypic level, on which then potentially natural selection can act. However, if you think about it a little more closely, you realize that this is actually a double-edged sword. Why so? Because the same recombination which can bring these alleles into favorable combinations, they can also break the same favorable combinations later, right. Now, therefore, when particularly let us say you have a very stable environment wherein you have ended up getting a nice beneficial combination which is increasing the fitness, you do not really want that thing to be broken down. If that combination gets broken down, then obviously after some time it is not there, and therefore, retaining that combination becomes very important. Now, that means that under stable environment, this thing generation of novel genotypes due to recombination, that perhaps is not going to be a potent force in terms of maintenance of sexual reproduction. However, what really happens if the environment is not stable? What happens if the environment is fluctuating, or what happens if the environment is changing constantly, not necessarily fluctuating, but changing in a particular direction? Under all those situations, the ability to continuously generate new variation, even if some of the old beneficial variation might get lost, that might end up being a favored trait. And there is one particular condition where it can really become a favored trait, and that one particular condition is in the context of what is known as the Red Queen hypothesis. What is that? So, we know that we are always surrounded and by all kinds of pathogens and parasites, and not only us, all organisms are surrounded by pathogens and parasites. Now, these pathogens and parasites, they typically have a very short generation time, because of which they evolve extremely fast, and because of that they typically end up evolving very fast to be able to invade or infect or use what is the most common genotype, right. And this, so basically they can specialize very fast on the common host genotypes. Now, if that happens, then if you have a genotype in the host which is relatively rare, then that can escape the infection by these specialized parasites, and if they can escape the infection, the fitness of that rare genotype is going to go up. And because recombination is continuously creating rare allelic combinations, therefore, this can become a mechanism by which a host can co-evolve alongside its enemies. So, basically what is happening is that let us say you have lots of host genotypes, some of which are very common, others are not so common, and now you have the pathogen is evolving extremely fast, much faster than the host, and therefore it is becoming specialized to whichever is the common genotype, and now if the host has a mechanism by which it can now generate rare genotypes, then the pathogens will not be able to invade them that easily, and therefore, that mechanism of creating a rare genotype is what is going to get favored, and recombination is one such mechanism. So, this overall picture was proposed by this evolutionary biologist called Leigh Van Walen in 1973. To be fair, he explained it, he proposed it to explain the phenomena of co-evolution when two or more species are evolving in response to each other. He did not propose it in the context of sex. However, very soon people figured out that this is a very nice potential explanation to the phenomena of sexual reproduction, and they called it the Red Queen hypothesis. Now, why did they call it the Red Queen hypothesis? This is named after the character of Red Queen in Lewis Carroll's book Through the Looking Glass. Now, in that book there is this character very famously tells Alice, the main protagonist of the series, now here you see it takes all the running you do to keep in the same place, which is a metaphor to explain the fact that in co-evolution two species are evolving as fast as they can and as much as they can, simply not to fall behind the other species. So, it is not that one is able to completely outwit the other, it is like an arms race. Country 1 makes weapon 1 and country 2 now has to make something better. Once it has made something better, country 1 has to make something even better, and so on and so forth. So, the relative position does not really change, except you keep on making more and more advanced weaponries. So, same thing is happening over here. Both the co-evolving species are evolving as fast as they can, yet they are not really able to conclusively beat the other species. So, the statement of the Red Queen hypothesis is that sexual reproduction in hosts is selected due to co-evolutionary interaction between host and parasites to reduce infection risks. Now, of course, you can see that this is a multi-step hypothesis. It is not a simple A therefore B kind of a thing. So, because the reasoning is multi-step, therefore, each component of this reasoning chain has been actually validated extremely thoroughly in the literature. So, this is a very well studied hypothesis. So, instead of giving you one or two validations, what I will do is I will show you what are the various steps in the reasoning, and I will also tell you where the validations have happened, and I am also going to give you references where you can go and check them out on your own. I mean, most of these steps have been validated multiple times in multiple systems. So, the total number of references is unmanageably high, but what is more important for us is to understand what the various steps are and whether they have been validated or not. So, here are the various observations. So, the first thing is, these are the references where you will find the primary references to all these things. So, the first thing is that it shows that this particular hypothesis is assuming that if you have sexual reproduction, it is beneficial in the presence of parasites, which tells you that if there are no parasites, asexual is what should be favored, and if there are parasites, sexual should be favored. And that is precisely what we see, that in nature, obviously there are some places where there are more parasites, some places there are less parasites. Wherever there are less parasites, the asexual reproduction mode dominates, whereas if there are more parasites, the sexual one dominates. So, it suggests that asexuals eliminate sexuals, whereas where parasite mediated selection is weak. Second thing is that for this entire thing to happen, the parasites need to be able to specialize, because if the parasite is able to infect all the species, all the hosts irrespective of the host genotype, then this entire thing does not work, the entire Red Queen hypothesis does not work. So, this will work if the parasite is able to figure out the most common genotype and is able to specialize for it, and turns out that parasites in many cases it has been shown can indeed do that. And not only that, not only can the parasite specialize, they can specialize against the common host genotype. That is also very, very important, and many experiments, lab experiments as well as field work for a wide range of taxa have explicitly shown that parasites can not only specialize, they will specialize against the common host genotype. Then fourth point is that when the parasite specializes, it should be able to specialize in a way that it can exploit the features of the common genotype, right. In other words, it is not just simple growth, it should be able to take care of something which the host has evolved, and in a variety of microbes, plants and animals, it has been shown that the parasites are capable of doing that. And fifthly, it has been shown that when you have hermaphroditic worms, okay, like the earthworms where the males and the females are present in the same body, in all those cases when you expose such hermaphrodites to pathogens, virulent pathogens, then the evolutionarily speaking the rate of outcrossing increases. Outcrossing is lack of selfing, okay, where the males of one organism explicitly the male gametes of one organism are fertilized by the female gametes of the other one and vice versa. So, in all those cases where the hermaphrodites are exposed to pathogens, that is where the rate of outcrossing goes up, and not only this, these outcrossed offspring, they are actually more resistant to the self-fertilized offspring, which basically means that this outcrossing is actually helping the host in order to fight the infection again of the pathogens, okay, much more than the self-fertilized offspring. So, the main mechanism that sex is going to lead to resistance to infection, that is what is being proven here. And finally, it has been shown in flower beetles, Tribolium, that if you have antagonistic coevolution, which means that where you have a pathogen and a host and the pathogen is actually harming the host and they are co-evolving, in those cases the rate of recombination itself can increase in these organisms. So, all these are various chains in the or links in the chain of reasoning for the red queen hypothesis, and individually each one of them has been very, very nicely validated. So, that is what I am saying over here. Almost every aspect has been very well validated in some system or the other. But if that be the case, that means the problem is solved. We know what is happening. And turns out that it is not that easy. Why is it not that easy? That is because although there are many, many cases where things have been supported, there are a few empirical examples where the various chains of the various links of the red queen hypothesis, they have not been supported. In other words, the support is not universal, and therefore today scientists think that although the red queen hypothesis by itself is fine, okay, having sex does help the organisms to combat infection, but it is not sufficient to explain the maintenance of sexual reproduction under all conditions. So, this is very nicely put by Sarah Otto and S. Nuismer when they say, I mean, after a very long theoretical analysis and with some experimental data also, I mean analysis of other people's experimental data, that we conclude that although the red queen favors sex under certain circumstances, it alone does not account for the ubiquity of sex. Okay. So, it is not a necessary condition for sure, and I mean when it happens it will explain sex, but there are so many conditions where you do not even expect this to be the main reason. Great. So, what else do we have? So, this leads us to our second hypothesis, but before we get to that second hypothesis, we have to understand a very major problem that happens in the context of asexual reproduction. What is it? So, remember that deleterious mutations are always appearing in a population. I mean, mutations are always appearing in a population, and as we discussed when we were talking about mutation, that most non-neutral mutations are actually deleterious. So, deleterious mutations are always cropping up. Now, when these mutations are cropping up in an asexual population, remember there is no recombination happening in them, right. Now, because there is no recombination happening, therefore, the healthiest individuals are the ones which have the fewest harmful mutations in their genome. Okay. So, suppose you have an individual which has no harmful mutation and you consider the fitness of that individual as 1, then obviously all the individuals which have 1, 2, 3, 4 whatever number of harmful mutations, their fitness will be less than that of the one which has 0 harmful mutation. So, this reduction in fitness is what is known as the genetic load. So, if you are talking about the healthiest individual, as I am pointing over here, these are the ones which have the fewest harmful mutation, and therefore, technically speaking, these are the ones which are the least loaded, okay, amongst all the genotypes that are present in the asexual population. Now, because of this, if there is genetic drift also happening in the population, then this least loaded class, although selection is favoring it because it has the highest fitness, but remember selection and drift always are working in tandem or always are working sometimes opposing to each other, and therefore, the least loaded class can be completely lost, particularly when population size is small, and in that case other less fit individuals can end up surviving in the population, right. When that happens, what is the consequence of that? Once the least loaded class is lost, there is no other way of naturally restoring this except back mutation, of course. There is no other way of naturally restoring this least loaded class in the absence of recombination. This process is irreversible, right. So, what is the outcome of this? The outcome of this is what you can think in terms of instrument very often used in by auto mechanics and others, what is known as a ratchet. So, what is a ratchet? A ratchet is something which you will be using, which you will be able to turn a nut or a socket in one direction, but you will not be able to turn it in the other direction, right. So, a ratchet mechanism is a mechanism which only turns in one direction and not the other. So, you have what we discussed in the previous slide. This is in technical language is known as Muller's ratchet, which is a process by which the absence of recombination leads to an irreversible accumulation of deleterious mutations in a population. Why is that? Because if you do not have recombination, then once harmful mutations accumulate, and if the least loaded class gets wiped out, there is no way of going back. So, the accumulation of mutation becomes an irreversible process in an asexual population. So, of course, big examples of this are bacteria, but a very human centric example is the human Y chromosome. Remember, we have X and have Y chromosomes, and the females are XX, the males are XY in humans, and the X chromosomes, they can recombine with each other, but since the Y chromosome does not have a homologous chromosome, therefore, the Y chromosome has not been recombining for a very long time, at least as long as our species has been existing in the XY form, and therefore, over time the Y chromosome of human males have actually ended up acquiring a very large number of harmful mutations, and people think that at some point the number of mutations will be so high that this chromosome will essentially kind of breakdown, right. So, this is a major example of Muller's ratchet happening in real life, and Muller's ratchet is named after Hermann Joseph Muller. So, Hermann Joseph Muller was associated with Thomas Hunt Morgan, Morgan's famous fly lab. He himself is a Nobel laureate for discovering X-ray mutagenesis, and as we are going to see, ended up making massive contribution to the field of genetics. Now, understanding this particular problem of what happens if you do not have recombination immediately tells us what will be the benefit of having recombination. So, the recombination will end up recreating the genotypes every generation, and therefore, if you have, let us say, genome, let us say one genome has one harmful mutation, another genome has another harmful mutation, recombination can actually create a genome which will not have any harmful mutation. In other words, the least loaded genotype, or the loading of the genotypes can actually go down because of recombination. You can end up getting quote unquote clean genotypes without any harmful mutations potentially every reproductive cycle. I mean, it does not happen every reproductive cycle, there are probabilities associated with them, but they can keep on, the process can keep on throwing these kinds of clean genomes. Now, if that happens, if you have the clean genomes, so to speak, and if you have other genomes in which many harmful mutations have come together, then that actually allows selection to purge the mutations, right. And if that happens, then the number of harmful mutations that are there in the population gene pool actually goes down. In other words, selection becomes a lot more effective in throwing out the harmful genetic mutations. So, I will give you one very nice empirical example for this, empirical validation of this. So, this is the New Zealand mud snail. I mean, the name is really a tongue twister, Potamopyrgus antipodarum. Okay, we will just call it the mud snail. So, this particular species is very interesting because it exists in two forms. It can exist both in the sexual form and in the asexual form, and sometimes in the same populations. We are talking about natural populations here. Okay. In the same natural population, it can have both sexually reproducing and asexually reproducing forms. So, what Nyman et al. Did in 2010 was they showed that the asexual lineages, they are experiencing a subsequently a substantially higher rate of accumulation of deleterious non-synonymous substitutions than the corresponding sexual lineages. In other words, the accumulation of harmful mutations is greater for the sexuals than the asexuals than the sexuals. Is this the only example that we have? Absolutely not, there are many more. So, I will give you one more. So, this is in the context of these stick insects, these are known as the Timema stick insects. Henry et al. 2012 showed that there was a 3.6 to 13.4 fold times higher rate of mutation accumulation in the asexual populations compared to the sexuals, right. So, this makes it a very nice strong mechanism for as the benefit of sex. So, another mechanism with for the benefit of sex is related to another issue with asexual reproduction, what is known as clonal interference. So, in this particular case, here is a diagram, we need to look at what this diagram is doing in detail. So, here let us assume that we have a 1 locus 2 allele scenario where the population is starting with a genotype small a small b. We have time on x axis, we have frequency on the y axis. So, this is color coded. Okay. So, the frequency of AB is white, the frequency of small a capital B is red, capital A small b is green, capital A capital B is yellow, right. So, for any given color, the length of the line along the y axis, that tells you what is the frequency of that particular type. So, for example, if you can follow my cursor over here, the entire thing is white, which basically means that the entire population is small a small b. Over here, this much, the white is this much, and you have this much is red, this much is green. So, this is what is telling you what is the relative frequency of small a capital B, small b and capital A small b in, let us say, this region, and so on. So, what is happening here? So, let us assume that this is a asexually reproducing population, and we are starting with the entire population being small a small b. So, the only way in which a new mutation can come in over here is through, well, a new allele can come in over here is through mutation. So, at some point there is a new mutation over here, and let us say the small b mutates to capital B, and after that let us assume that going from a small letter to a large letter increases the fitness. So, small a capital B has a larger fitness, and therefore, the frequency of the small a capital B keeps on increasing, right. That is what is meant by when this thing, the thing expands, the red color expands. Similarly, at another point over here, the capital small a allele in one individual, in one lineage becomes capital A, and this is where capital A small b starts, and it starts expanding. Now, what you have to realize is that in any asexual population, whenever you have a mutation happening, that is when a new lineage is starting, right. So, that is what I mean by saying a beneficial mutation forms a new lineage. Why does it have to be beneficial? Because if it is not beneficial, then by selection it will go out, right. So, it will not be able to expand. Only beneficial mutations, their lineages are going to expand. Great. So, now if you have multiple beneficial mutations like what we are having over here, then there can be several distinct lineages that can be formed, and that is what is happening over here. You have the, let us say in this range, you have small a small b, but you also have capital A small b, and you also have small a capital B. Now, when these mutations, when these lineages they are expanding, okay, these lineages they are also competing with each other, right, competing for survival, competing for reproduction. When they are competing with each other, if you look at the overall fitness of the population, that overall fitness of the population is actually getting reduced. Why? Because let us assume for a moment, let us say that capital A small b is more fit than either small a small b or small a capital B, right. So, if the entire population were composed of capital A small b like what it is over here, then the fitness of the population will be maximum. But because these guys, the capital A small b guys are competing with small a capital B and small a small b, and therefore, all three of them are existing together over here, therefore, this is going to take some time to reach to this, this stage where you have only one genotype. So, that is what we mean by saying that the rate of increase of fitness, how fast capital A small b is able to get rid of the other two, that rate of increase of fitness, which basically means rate of adaptation, that rate of adaptation is what is going down because of the competition. And this also slows down the fixation of the beneficial mutation. Fixation is, remember, when frequency goes to 1, right. So, this is where the fixation happens, but because of this competition, the time that is taken to get to fixation, that is reduced. Now, here you have a stage, let us say, where capital A small b has happened. Now, remember, capital A capital B is the fittest phenotype. Now, the only way that you will go from capital A small b to capital A capital B is you have to wait till the small b mutates to capital B in one of the asexual lineages. And when that happens, in this case let us say that happens over here, after this point again capital A small b is going to compete with capital A capital B, and therefore, going to this final capital A capital B is going to take quite a bit of a time. In other words, and sometimes what happens is in this process, let us say an allele which otherwise could have complemented with another allele to give the fittest phenotype, that might even get lost during this process when it is competing with others. And that is what I mean by saying that mutations that become fixed are from the most successful competing lineages, but that does not necessarily mean that the final product that you get is the combination that would have actually given you the highest fitness. In other words, this fact that you are having competing lineages containing various mutations, that can end up not only slowing down the rate of adaptation, that can also end up putting constraints on whether you are able to attain the genotype with maximum fitness or not. So, this particular stuff, this is what is known as clonal interference, and the definition is that the competition between lineages arising from different beneficial mutations in an asexually reproducing population. So, clonal interference is simply the competition part of it. The outcome of clonal interference is slowing down the rate of adaptation. Now, I have taken this definition from this particular paper, but that does not mean that these were the people who propounded it. Actually, this was proposed by Hermann Joseph Muller in 1932, the same Muller that we talked about a few minutes back. And turns out that clonal interference actually plays an enormous role in the evolution of microbes and viruses, and it is also extremely important in the dynamics of cancer cells. So, if you remember the way people have been thinking about treating cancer, in fact the way cancer is treated even now is that you do chemotherapy or you zap them with lots of radiation and you end up killing everybody, and this kind of a thing can often lead to relapse. So, one of the stuffs that people figured out is that because cancer cells are reproducing clonally, there is a lot of chance of this kind of clonal interference and therefore competition among the cancer cells. And hence people actually said that why do not we use this competition amongst the cells to keep the growth of the cancer in check. And that is why one of the lines of therapy that is now people are, I think it is in clinical trial, is known as adaptive therapy, where the whole idea is do not kill off all the cancer cells, particularly do not kill off the competitors, identify where the competition is happening and do the treatment in such a way that the competing cells, by competing with each other, actually end up keeping the cancer, overall cancer, in check. Okay. This is, as I said, is leading to new treatment possibilities, and people are actively trying to see if one can manage cancer in this way. The other massive example of this kind of competition, clonal interference, happening is in the context of what I am sure all of you are aware of, was the how COVID virus evolved during the pandemic. So, if you remember all these terms, alpha, delta, omicron, etc., these are what? These are different lineages of the COVID virus, right. So, if you remember, the first human infection happened somewhere around late 2019, the Wuhan in China, and then these early strains, they kept on evolving, they kept on acquiring mutations. Remember, these are asexual lineages, right. So, they acquire mutation one after the other, and then at some point there was a branching over here, and at some point we got to what is known as the alpha strain. So, this is where these are the alpha strains, and they started increasing in number. So, if you look at this graph over here, initial phase, the first early strains were evolving. At some point alpha came in, and alpha was more competitive than the others. So, alpha increased in frequency greatly very soon. But while alpha was increasing in frequency, at some point from the early strains we got the delta strain, and then delta strain came in somewhere over here, and delta was much more competitive than either alpha or the early strains, and therefore delta moved very fast. Delta frequency increased extremely fast. You can see that very soon these two are declining very fast. This is where we have our second wave, and then while delta was evolving, at some point you got omicron from the other branch, and omicron turned out to be even more competitive than alpha, and once omicron came in, then actually it ended up just swamping the entire stuff all over the world, and very soon all we had was Omicron, right. Fortunately, Omicron was a lot less virulent than Delta, and therefore, although it caused a lot of death, it was relatively less. But the main thing that I want you to understand is that if COVID-19 were reproducing sexually, then the rate at which Delta and Omicron evolved, that would have happened much faster. In other words, we would have gone into this high level of virulence and probably even higher level much faster than what it actually ended up, which was blindingly fast to begin with. So, what happens in a sexual population? So, here I am showing the asexual graph all over again. It is the same graph that we discussed a few slides back, but I am now discussing this in the context of the sexuals over here. So, the first part is the same. We start with small a small b in both cases. In both cases, the capital A small b and the small a capital B, they appear roughly around the same time, but what happens after that is the interesting part. Initially, they will expand on their own because probably they are in very different parts of the population, they are not coming close to each other. But once these genotypes come close to each other and they mate and then recombination happens, then this formation of capital A capital B is going to happen much sooner. And once that happens, this is also going to spread much faster, and therefore, due to recombination, multiple mutations that have arisen in different backgrounds, they will be able to come on the same into the same genome much faster, and thereby reducing the competition. So, here the only way you will go from small a small b to capital A capital B is if the two mutations capital A and capital B arise one after the other in the same lineage, otherwise you can't get here. But in this particular case, these mutations can happen, capital B mutation happens in this lineage, capital A mutation happens in this lineage, and then recombination brings capital A capital B into the same genome, which is what is happening over here. So, this ability of recombination to bring mutations that have happened in different backgrounds into the same background, that is what is speeding up the overall rate of adaptation in these populations. Now, which simply means that sexual populations can adapt much faster. Do we have okay, sorry. So, this is the formal statement of it. This is what is known as the Fisher-Muller model. It simply says that advantage of sex results from recombining competing beneficial mutations into one lineage. As you can see, named after Ronald Fisher and Hermann Muller, the same Hermann Muller that we talked about. So, you can see he made serious contributions into the evolution of sex field, and interestingly, this is also known as the Viker of Bray hypothesis. Now, what does that mean? What is a Viker and what is Bray? So, Viker is a kind of a priest in the Christian church, and Bray is a place in England. So, Viker of Bray is actually a fictional character who essentially he was basically adaptable. So, this particular person, so the story goes, when the monarch of England changed, this person changed his religious belief, and when people actually questioned him that how can he be like that, he simply says that, look, I have only one goal in mind, which is to remain the Viker of Bray throughout my life, and in order to do that I am adaptable along other axes. So, this is obviously a very tongue in cheek funny reference, but here the whole idea is that the sexual reproduction is able to bring together all kinds of benefits and make the most adaptable genotype possible, okay, the most beneficial genotype possible simply by putting together things that have or mutations that have happened in very different lineages. Now, do we have empirical reason to believe that this works? Turns out that we do. So, I will give you one very famous study on this. This was by Tim Cooper in 2007, and here he actually ended up using E. coli. Now, this is very interesting. E. coli is a bacteria, right. So, normally you expect that this is going to reproduce asexually, which it does, but turns out that in E. coli there are these things known as F plasmids, right. You know plasmids, these are circular pieces of DNA, extrachromosomal DNA. So, these F plasmids, they actually end up promoting exchange of genes across E. coli cells, and in that sense there is exchange of DNA, and this is mimicking recombination essentially. It is not a regular recombination in the sense of exchanging homologous parts of homologous chromosome, but it is actually at least exchanging genes. And he, of course, as a control, he also had those E. coli which did not contain the F plasmids, and therefore they were evolving asexually. So, what he did was he took these two kinds of E. coli and he engineered this in a background where the mutation rate was high. How can you do that? So, turns out in E. coli you have certain genes, mutase, mutal, etc., wherein if you put in some mutations in those genes, then the rate of mutation in the overall genome can go up quite a bit, tenfold, 100-fold and so on and so forth. So, he engineered one of those hypermutation things, and thereby the background on which this F plus F minus things is happening, the background had a high mutation rate. Now, if that be the case, if the mutation rate is high, what is happening? You are getting lots and lots of mutations, right, many of which are also going to be harmful. So, now the question is about combining of beneficial mutations. If everything goes all right, then if the Vicar-of-Bray hypothesis is correct, then you expect that the ones which are containing the F plasmid, the ones which are having recombination like phenomena, they are the ones who will be able to access the beneficial mutation faster, and thereby they are the ones who will be able to increase their fitness better. And that is pretty much what he ended up finding. So, he showed that the bacteria which is containing the F plasmid, those lines, the recombination actually ended up increasing the rate of adaptation about 3-fold, and this was due to a beneficial mutation. So, using sequencing he was able to identify precisely which mutation led to this increased adaptation, and this is the best part, he showed that in the F minus lines, in the absence of recombination, this mutation was taking much longer to fix, and over the course of its substitution, it ended up conferring a much reduced competitive advantage. In other words, when you have absence of recombination, then the competition among the various genotypes was much greater compared to the situation where you had the recombination. Interestingly, this entire thing works if and only if the mutation supply is high. If the mutation supply was low, which is what he did by having another set of lines where the mutation, he did not have the mutase allele and therefore the mutation supply is low, in that particular case there was no difference in adaptation. So, basically what he ended up showing was that recombination increases the rate of adaptation, correct, but it does so only when there are competing beneficial mutations in the population. If you do not have competing beneficial mutations in the population, then recombination is not going to play a role in terms of increasing the rate of adaptation, which makes a lot of sense, right, because that is the context in which the entire hypothesis has been put, that this will work if and only if you have competing beneficial mutations in the population. And this also tells you about the weakness of the hypothesis. I mean, hypothesis is fine, but rather the specificity of the hypothesis. Okay, this will only work under that particular scenario where there are competing beneficial mutations. So, where are we? We today have a very good understanding of the benefits of sexual reproduction, and all these benefits theoretically proposed, they have been extensively empirically verified, they work. Now, remember I gave you 3 hypotheses, but these are not all the hypotheses that we have there. There are many others. So, for example, you have the so called best man hypothesis, you have the tangled bank hypothesis, you have the mutational deterministic hypothesis, and so on and so forth. There are multiple. Many of those hypotheses, almost all of them, have some empirical validation or the other. But all the hypotheses, they require specialized conditions, like what we discussed in the context of these 3. And because they require specialized condition, there is no single hypothesis that has led to theoretical models that can confidently account for the appearance of sex over a wide range of biological scenarios. Okay, this is the point. We have a bunch of hypotheses, all the hypotheses make sense, but all of them require specific scenarios. If those specific scenarios are there, they will work, but then there is no one thing that is explaining it across the board. Okay, nothing is explaining on its own that why 99% of the animals are sexual. In other words, we do not have a general theory. Now, is this a problem? Because in biology, we hardly have any general theories, right? So, lots of people have argued that the lack of a general theory is not that big an issue. But there are other people who suggest that 99%, that is too big a number. There should be something that is kind of more overarching. And now that is a article of faith, that is a belief that there should be such thing. It is like the theory of everything in physics. People are still searching for the theory of everything. That does not necessarily mean that all the specialized theories that we have, they do not work, they do. People are still searching. So, this is what we understand about why sex as of this moment. But one thing that we really understand about sex is its implication in terms of explaining certain very, very strange patterns in the context of biology in the field what is known as sexual selection. Okay. What happens when one sex ends up choosing for certain characteristics in the other sex? What does it lead to and what kind of peculiar patterns does it explain? That is something that we are going to discuss in our next discussion. See you till then.